Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about amino acids and peptides as an introduction into our discussion on proteins. When amino acids are joined together by dehydration synthesis, the result is a kind of molecule called a polypeptide. Now, polypeptides and proteins are a little bit different in the sense that proteins are fully folded, three-dimensionally shaped molecules. The importance of folding and shape in protein structure and function is something that's a little bit unique and is something that's not quite as necessary when you're talking about things like carbohydrates and nucleic acids, other types of macromolecules. So the idea here is similar to what we have seen before. Amino acids are the monomer on which we are going to use to make our polypeptide polymer. So when we join these amino acids together by dehydration synthesis, we forge a type of covalent bond called a peptide bond in between those adjoining amino acids. And in doing so, we start to build a peptide. So when we do this over and over and over again and we build this amino acid polymer to be a decent length, we now have a polypeptide, and this polypeptide will then be capable of folding into a three-dimensional structure, and the result of that folding will be a functional protein, or at least we hope. So let's take a look at amino acid structure. Every amino acid has four essential components to it. First, highlighted in purple here, you have the alpha carbon. This serves as the center of the amino acid, and all of the other components of the amino acid will be attached to this carbon as you see it here. In green, you see what is called the alpha amine. This functional group is attached to the alpha carbon, and it participates in the formation of peptide bonds. If it is not actively tied up in a peptide bond, it will have a positive charge at a neutral pH because this is an amine functional group. In red, you see the alpha carboxyl group, which is also attached to the alpha carbon. Just like the alpha amine, the alpha carboxyl also participates in the formation of peptide bonds, and if it is not tied up in a peptide bond, it will be negatively charged at neutral pH values. So far we have introduced three of the four components of an amino acid. Every single one of the 20 amino acids that are used to build our proteins have these three components, and there's nothing different about them. However, what's going to separate one amino acid from the other 19 that we use to build proteins is what we call the side chain, which is highlighted here in orange. The side chain is just a functional group that will impart some kind of unique property to each amino acid. Seeing the letter R here, you might be tempted to think that R stands for some element in the periodic table, but it does not. R in this case just stands for the remainder of the molecule. R could refer to something as small as a hydrogen atom or something as big as a very long hydrocarbon chain. So when we actually form a peptide bond through dehydration synthesis, the peptide bond will be forged between the alpha amine of one amino acid, which you can see right here, and the alpha carboxyl of a different amino acid. Highlighted in blue here, you can of course see the element of water that is going to be lost from these two molecules when we do the dehydration synthesis. So after dehydration synthesis, you can see the element of water has been lost, and in red you can see the peptide bond that has now been forged. This peptide bond is actually very interesting because of how strong it is. It is about 1.5 times stronger than your typical single covalent bond. I won't get into the nitty-gritty of it, but you'll notice that this peptide bond is very close by to this double bond right here, and it turns out that the double bond can share some of its properties with this peptide bond. So that results in the peptide bond being stronger than you would expect and being especially hard to break, which helps to give proteins a lot of extra stability. One thing you will notice about this dipeptide, two amino acids joined together, is that this dipeptide still has a free amine highlighted in green and a free carboxyl highlighted in red. If we add a third amino acid onto this dipeptide to turn it into a tripeptide, you'll notice that the tripeptide still has a free amine and a free carboxyl. So this is going to be a little bit of a theme for us. No matter how big or how small a peptide, a polypeptide, or a protein is, it is always going to have what is called an N-terminus, one end of the protein, and a C-terminus, the other end of the protein. I have illustrated the N-terminus and the C-terminus as being electrically neutral, but remember, both of these functional groups will be electrically charged at neutral pH values. So finally, the last thing I want you to notice about this discussion on peptide bond formation is that the side chains, designated as the letter R, do not participate in the peptide bonds, but rather they just kind of hang off to the side. So when we form a peptide bond, it is happening between the amine of one amino acid and the carboxyl of another amino acid. The side chains do not get involved in peptide bond formation. Okay, so we mentioned that there are 20 different amino acids that we can use to build proteins in the cell. What we're going to do here is we're going to classify those 20 amino acids based on the chemical properties of their side chain functional groups. Classifying amino acids based on their side chains makes the most sense since the other three components of the amino acid are pretty much identical between all of the amino acids. First, we have amino acids that have nonpolar or hydrophobic side chains. You have amino acids that have polar hydrophilic side chains. You have amino acids with acidic or basic side chains. And then finally, you have amino acids with aromatic side chains. We'll talk a little bit about what that term aromatic means, but basically what it's going to essentially mean is that these side chains are very, very large and very hard to fit into tight spaces. Okay, so we'll start off with the nonpolar hydrophobic side chains. And before we go any further, no, you do not need to memorize any of the amino acids. You don't need to memorize their chemical structures. You don't need to memorize their side chain composition. You do need to be aware of the classes of amino acids. So here you can see 4 examples of the nonpolar hydrophobic side chains: glycine, alanine, valine, and leucine. And if you look at the side chains, which are highlighted in orange here, you'll notice that there is a common theme here: carbon and hydrogen. We've mentioned before in several videos by now that anytime you have a lot of carbon and hydrogen, this is going to spell nonpolar chemical properties, meaning that these side chains will not interact well with water. So these are not the only 4 nonpolar side chains. Some other examples are isoleucine, methionine, proline, and cysteine. But the main takeaway is that these side chains, as the protein folds, are not going to interact well with water, so they will want to steer clear of any water-rich areas. Next, we'll move on to the polar hydrophilic side chains. Some examples here are serine, threonine, and asparagine. And what you'll notice here is that these side chains include polar functional groups. Hydroxyl groups in the case of serine and threonine, and then a polar amide functional group in the case of asparagine. Glutamine will actually also be very similar to asparagine, but I couldn't quite fit it onto this slide. So since these side chains either have polar hydroxyl or polar amide functional groups, they can form hydrogen bonds, and that's going to mean that they will interact well with other polar molecules, and that, of course, includes water. But be aware, though, these side chains are not fully charged. As polar functional groups, they will only have partial charges. Next, we'll move on to the acidic side chains, which of course are going to be negatively charged at neutral pH values, and these include aspartate and glutamate. So these side chains, because they include carboxyl functional groups, they of course, as I said, will be negatively charged at physiological pH values. They will interact well with water, but there's actually something they would probably rather do, and we'll talk about that in the future. The basic side chains, which are positively charged at neutral pH values, are going to include amine functional groups, which we have seen before carry positive charges. These include lysine, arginine, and histidine. So these side chains, because they have amine functional groups, they will be positively charged at physiological pH values. Just as the case was with the acidic side chains, these basic side chains will also interact well with water, but again, there is a different type of interaction that they would probably rather participate in. And then for the aromatic or bulky side chains, these are similar to what we saw before. These are going to mostly consist of carbons and hydrogens, and that of course will make them hydrophobic and adverse to interacting with water. A good example of one of these you can see here is phenylalanine. So the word aromatic in this case refers to these closed ring structures like you see right here. So if you see an amino acid side chain that is a ring in its composition, that's one of the aromatic ones. Some other examples include tryptophan and tyrosine. And if you've noticed, every amino acid not only has its full name, but also a single letter abbreviation. You do not need to memorize those abbreviations, but it's good to be aware of them, knowing that you can and often will abbreviate amino acids with just a single letter. So we mentioned that these side chains, because of their heavy hydrocarbon composition, they are nonpolar, they're going to be hydrophobic, they're not going to want to interact with water, but because of the sheer size of these side chains, not only will they not want to interact with water, it's going to be a little tough to fit them into tight spaces when the protein eventually folds. Okay, so as the protein starts to fold into its final three-dimensional structure, side chains are going to be forced to interact with one another and with water that's in the environment. It's often helpful to think of a protein as being something that's just floating around in a bodily fluid. So on the left, you can see individual amino acid monomers with their side chains hanging out, and on the right, you can see those same amino acids attached together with peptide bonds, which are highlighted in yellow, and you can see that different types of side chains are going to need to be positioned in different ways to give us favorable interactions between the side chain functional groups and other functional groups or with water. Okay, that's going to do it for this video. Our next video is going to get further into protein folding and satisfying the ways in which these side chains interact with other side chains and with the environment. But we'll go ahead and close this video off here for now. Here is a list of vocabulary terms you probably want to be aware of. And for checking your understanding, number 1, what are the major components of an amino acid? Number 2, how are peptide bonds formed? Number 3, how are amino acids classified by their side chains? And number 4, how does the order of amino acids affect protein folding? Number 4, you may want to go ahead and watch the next video in our series before you really start thinking about that one. Okay, that's going to do it for this video. If you have any questions, go ahead and drop it in the comments section, and I will see you for the next video.